Now we have another presenter from the University on Montpellier. I tried to read, I apologize for wrong pronunciation, Merouane Elazami Elhassani. And his talk should be about identifying the part of exons of immunoglobulins and T cells receptors. Please.
Okay, do you hear me? It's okay.
Yes.
I shared my screen. It is visible.
Yes, we can see it.
Okay. So firstly, I would like to thank you for your conference and your organization, because it is very, very interesting to participate in this kind of conferences and get the exchange with people, know other topics. My topics is a little bit new, so I'm trying to apply artificial intelligence for immunogenetics. Especially in this talk, I'm going to talk about predicting the L part 1 region exon using deep learning. So first of all, my name is Merouane Elazami. I'm doing a PhD within EMBnet laboratory, the same as the previous speaker, in University of Montpellier, which is found, this PhD is found by Atos company. My background as a software engineer, so I'm trying to use this skill also to enhance bioinformatics. So in this talk, I'm going to talk about an introduction to my subject and talk about artificial intelligence and deep learning especially. And I'm going to talk about how we use it within EMBnet, which is EMBnet case, and future works and future perspectives, and a conclusion. So first of all, as you know, sequencing technology, we have witnessed they have advanced in recent years, and this comes with its own benefit, also with its own challenges. So as you know, today, when we are talking about genomics, which means also a big data science, and a huge amount of data were sequenced with a cheap price, and to keep up with all those the data, we need new tools to be able to analyze and annotate in our case, for example. So for that, in my PhD, I'm focusing on artificial intelligence. To be brief, artificial intelligence is a branch of computer science that tries to mimic human intelligence. In my case, I'm using a deep learning approach, which has proven its potential recently in many fields. So this deep learning approach is basically using a neural network. What is a neural network? A neural network is a structure that tries to mimic the human brain at some extent, and it is what is called the cornerstone of it. It is a neuron. A neuron simply tries to mimic the human neuron, which is found in the human brain. So mathematically expressed, a neuron is just simply a function that takes inputs and has an operation within it and tries to produce an output based on an objective goal. For example, the objective is to approximate a Y, for example. To see the difference between the deep learning approach and sequence alignment approach. So here the general pattern or general approach that normally we use, for example, supposing that we have a new sequence and we want to see the similarity and the functionality of this sequence. So the major approach is that we plug this sequence inside a tool, for example, and this tool tries to query a database to see the similarity and do alignments for the already existing sequences, and it gives us a certain result. But the deep learning approach is totally different. For example, instead of just querying a database, this database is used to extract some data sets. Those data sets are used to train a deep learning model. And these deep learning models try automatically to extract the meaningful features to do the classification. After the training comes the phase of evaluation. So this phase, we plug in, for example, the given sequence, and it tries to predict what it is. So the major concept here is that we learn from the past to predict the future instead just of querying a database. So in this case, in EMBnet case, here we are trying to use this artificial intelligence, especially the deep learning approach, to make or to train models to recognize the components of Ig antibodies and T cells receptors. For example, this is the general aspect that we want to reach, is that to be able to train AI models to be able to predict, for example, the V gene, the green region, the red region, the D gene, and the G gene, and the C gene. Those are the components for antibodies. So being able to predict correctly these components will enhance the annotation of them. So firstly, what we have done is that we have trained a naive deep learning model that is able to classify or to predict the components of the V region. You see that the V region is the green part here. This V region is composed of different motifs, of different components. So we trained a deep learning models so to classify if a given sequence belongs to this component or the other components. So how we did that is that we have used the EMBnet database, which is a LigM database, which is a database that contains annotated immunoglobulins, the immunoglobulins and T cells receptors. And we constituted or made some data sets, and those data sets, we shuffled them with random sequences in a manner which is called a supervised way of training. So the results are very interesting. So we have used 100,000 samples for the training. 60% of them were used for the training, 20% for the testing, and 20% for the validation. So we have reached almost 98% accuracy of predicting 16,000 samples, which the machine didn't know before that, or didn't see, sorry. So, and the loss function is 4%. So in this same approach, we try to go from the big component to the small components, bottom-up approach. So we have tried to train another model to predict only the L-part1 exon. This exon is the first exon located in the V region. So as we did in the before, but this kind of classification is a binary classification. We did the extraction from the database. We did trained a simple model, and we tried to learn if it is an L-part1 or not. So the results were also interesting, as you see, a 98% accuracy. So, but this model misses the motifs. So we needed another kind of model that tries to explain why the results were like that. So for that, we trained another model called the convolution neural network. This model has the particularity to extract the features to explain after that. So as you see here, the difference between the first model and the second model is that the second one extracts the features, and those features will help us to understand why the prediction was right or wrong. So the results were also interesting. So which means that there is some kind of similarities or some kind of features inside the data set that the model that learns from it and classify correctly the given sequences. So, but those two previous models, they have some issues. For example, the limit or the length of the sequence, because we cannot give a lengthiest sequence to those models. Another issue is that when trained, those models they don't take care of the position of the nucleotides, so the temporal features is ignored. So for that, we needed to look for another approach or to look for other models to be trained to be able to resolve those issues. So here comes in the first solution. So what we did is that we used a recurrent neural network combined with embedding. So we formed a kind of dictionary using the nucleotides. Those nucleotides are given as an input, and we can give the, whatever we want as a sequence, and it is decomposed as a camera sequence, then forms a dictionary, then gives us a classification. So, sorry for that. So also, if you notice that this model is more efficient than the other one, the previous one. So it takes just 4 epoch to be trained, and the results were really stable and really interesting. And to resolve the other issue, which is the position of the position of the nucleotides, which means the features where it is located. So we combine the 3 or the 4 architectures. So the first one here, the pipeline, the first one is the embedding for taking care of the limits of the sequence. The second one extracts the features. The third one takes care of the order of those features. And the fourth one tries to classify correctly the given sequence. So for the feature projection, here is the feature projected in 3D space to see what are the relevant features for the predictions. And the similarity is calculated based on the PCA distance. So we see what are the relevant features found in within the sequences that we used for training this model. So the implementation was done using TensorFlow, Keras, also Python, and the data processing and data cleansing was done by Panda library, also a Python library. And further work, which is need to be investigated, is that locating the component within sequences. So, for example, if this model, we give it a longer sequence that contains many components, the model tries to predict the whole bunch of sequences, not just a part of the sequence. So I need to figure out how to split the sequence to be able to locate and classify correctly the components, even there are many. Also the explanation based on the feature, which is generally a big issue within deep learning approach in general, not just in immunogenetics. The data augmentation, as you know, that deep learning is data hungry. It requires a lot of data. And in the database that I use it, we already have annotated a lot of T cells and immunoglobulins, but I think it is not enough to reach another level for this data, for the training the model. So in this branch, I am trying to explore what is called generative adversarial neural network to try to synthesize, to create myself those regions, if it is possible, using also this deep learning approach, and also to be able to predict all the components within the V region and the other components within the other regions. So as a conclusion, deep learning has demonstrated a big potential in other fields, also in immunoglobulins, immunogenetics, sorry. Deep learning has a potential for annotation because it can reduce the time for annotating. For example, if I give it a known sequence and the model is able to classify correctly the components, so annotation will be easy. And like I said, deep learning requires a huge amount of data, but also further investigation needs to be done, and also more studies need to be to prove the concept, which is really interesting. So I would like to thank you for your attention. I would like also to thank the University of Montpellier, the CNRS, the IMGT laboratory, Atos company, and also EMBnet for this opportunity to speak about new technologies and how we can collaborate together. Thank you so much. If you have any question, I will be ready to reply. Thank you.
Thank you very much for nice and very clear presentation. Now question. Domenica, do you have a question? I see rising hand.
Not really a question, but a comment. First of all, my best compliments for the clarity in the exposition of this work, but also for the work, because it is really interesting. An approach that show how much big is the potential of machine learning techniques in investigating the biological sequences. But I have only a question, and I am not, I do not work on annotation of non-coding RNA into genomes, but I know that this is not a trivial task because of the sequence characteristic of non-coding RNA. And my question is if I think that this approach could be, for example, exploited for this kind of analysis. Have you thought to this possible application? Because this would be really interesting.
So what was the question, sorry?
If you have thought to apply this type of approach also for the identification of non-coding RNA in genomes, because annotation of non-coding RNA is really difficult because they do not have so structured features like the protein coding gene have.
So yeah, to answer, as a quick answer, it is possible because this approach, it learns automatically. It is a data-driven approach. It learns from automatically the data. So it is us we need to define just the problematic. So for example, if we speak about non-coding region, so we just need to give the machine a samples, for example, samples of kind of non-coding regions, and to tell it in a supervised manner, this is a non-coding region. Can you figure out through your training what are the major motifs or the patterns that governs this non-coding region? So after being trained, so we can see if the machine is able really to recognize those regions and if it has generalized kind of knowledge, because the main difference here is that we use the data to extract the knowledge, and this knowledge is used to predict future results. So to answer, it is possible for sure. So also we can collaborate if you want on this subject. Thank you.
Really interesting. Yes, indeed. I think that I will contact you.
Yeah, thank you so much. Welcome.
Next questions. We still have some minutes.
Yeah. Any questions?
There are a few comments congratulating you.
Welcome.
So far, no additional question. Of course, you can always contact the presenter later if you will need any other. The information is available. So if we have no other questions, thank you very much again, Merouane.